In this video, we'll look at Mendelian inheritance patterns, focusing on a test cross. So what do we use a test cross for? It's used to determine the unknown genotype of an individual expressing a dominant phenotype. We would cross that individual in question, so the one that we're wanting to know about, with a known individual that is homozygous recessive. And then we can look at the results of the offspring to determine what we think the unknown individual is. Is that unknown individual homozygous dominant or are they heterozygous? Let's use lab coat color as an example. Let's say that we're dealing with the B gene, so this theoretical B gene here, and we know that this is controlling the lab coat color. Chocolate labs or brown labs are homozygous recessive little b, giving them that brown coat color. Whereas black labs, right, they could be a homozygous dominant big B. Because they're carrying that dominant big B allele, they would express the black coat color. However, a lab could be heterozygous little b. As long as they're carrying this, they're still going to have a black coat color. So the question is, are they homozygous dominant or are they heterozygous? Right? We could look at the resulting offspring to determine that. So if all the offspring come out to be black, then we would expect that that individual is homozygous dominant. Whereas if they come out in this proportion, right, they could be black or chocolate, aka brown, we would expect that that individual in question is heterozygous, big B, little b. Let's look at an example. So we know that, again, this individual right here has to be homozygous recessive, little b, little b. So we'll go ahead and write the chocolate labs genotype in the Punnett square like this. Now we want to know, is the individual in question homozygous dominant big B, little b, or are they heterozygous big B, little b? So all we would do is complete the Punnett square. So again, across and down. Right? That could be one scenario. If this is the case, all the offspring are heterozygous big B, little b, which means that they're carrying this dominant big B allele, which codes for black coat color. So we would expect every time that these individuals to mate, they would produce black coat colored puppies. What if the scenario is over here? What if the individual is heterozygous big B, little b? What happens when we cross it with a chocolate lab that's little b, fill in our Punnett square. Again, you're looking at cross and down. Now it comes to be something like this. Half of the offspring have the genotype big B, little b, and half of the offspring have the genotype little b, little b. We would expect half of the time the offspring would be black puppies, and then half of the time they would be brown puppies. So again, if we want to know what this individual is right here, we cross it with a known homozygous individual, and then we just look at what are the offspring? What do they come out to look like? And it will help us determine which genotype the individual in question has.